Okay, good morning everyone. Before I move on to my presentation, first of all, I would like to thank Sofa and Biosciences for this great opportunity to speak here about our research. This study is about studying an effect of the neuronal stimulants in phospholipid bilayers or the cell membrane. So next 20 minutes, I will be introducing you this cell membrane, neuronal stimulants molecules, their structure, activity, and also how we approach to study this effect of these neuronal stimulants according to our experimental strategies. And also, at the end, I will give you an biophysical insight into permeation process of these small molecules. Quickly, I will tell you some little information about cell membrane. So we already know that the cell membrane, it has a complex biological structure which is made up of phospholipid bilayer. So these are showing some of the examples of lipids structures. So you can see there's these hydrophilic head groups and also the hydrophobic tail groups. And the self-assembly nature of these phospholipids, they make this hydrophobic center area region and also hydrophilic extracellular and hydrophilic intracellular. So this is not these are not just hydrophobic and hydrophilic regions. These are just like sharply defined domains of hydrophilic and hydrophobic regions. So the stimulant molecules, they are type of drug that they increase the certain level of chemicals in our brain. And if you see some of these examples, you can see they have similar kind of structure. I will tell you why in next slide. And these are like specifically small and lipophilic molecules. And so the reason when we talk about these their molecules and its structure, so the first thing is these drugs, they should be hydrophobic enough to cross the lipid bilayer, and they should be reasonably hydrophilic enough to bind to their receptors, and also they are smaller in size. And if you take this example, so they have this hydrophobic ring and also hydrophilic nitrogen tail part. So and also I mentioned these especially confined hydrophilic and hydrophobic regions. So because of these structural and chemical specificities, we thought that we should give these a try, because they tend to interact with the lipid bilayer. So that's why we choose to study this system. And these molecules, these small molecules, they permeate into the lipid bilayer rapidly, easily they diffuse. So depending on several factors, and it's technically difficult, but there are techniques to study patch clamp and some of the fluorescence studies and computer simulations. So far we know that these molecules, they bind to the receptors, and they interact, and they release these neurotransmitters. And also there are studies showing then they disrupt the synaptic vesicles, and there are studies showing the hypothermia, calcium changes, accumulations in the rat tissues, but still the molecular level details of their behavior and the mechanism, how they interact with the lipid, the effect of these drugs on the living membrane, it's poorly understood. So that's why one reason we did this research. So this is summarizing the experimental strategy and the multi-technique approach this study. So the first question is how we can get the cell membrane. So we can get the natural cell membrane by using a cell line, or we can synthesize the cell membrane as like planar lipid membranes, and also we can computationally model the cell membrane. And by combining these fluorescence microscopy techniques and patch clamp, we can continue this study. And also by combining computational methods, what we really like trying to do here is to measure the real-time electric current activity, the permeability changes, so we can know the permeability changes, how they diffuse, and also especially from these computational studies, we can get the molecular level behavior of these molecules in the cell membrane, and we can get the energies, we can study the structural and dynamics of these cell membranes with the effect of these methamphetamines. And so a little more about the lipid membranes. I already told that these we can get the natural lipid membrane. So in this study, we are using H22 mouse hippocampal cell line, and also they actually mimic this central nervous system environment because they have this complex composition with the intact cell context. And the planar lipid bilayers, they also mimic the cellular environment but with the control composition. Sometimes it's good to have this reduced complexity, because sometimes if we want to study a system without getting influenced by other factors, so it's good to we can use these simpler systems. So there are like several types. The supported lipid bilayers, it could be on a solid surface, or it could be on a liquid surface. And also there are black lipid membranes. Both these black lipid membranes and the lipid vesicles, they have this accessibility to approach both sides of the lipid bilayer. And also we can use these black lipid membranes. They have like, we can simultaneously measure the electric current measurements and also the microscopic measurements. So these are summarizing like the ways that we can prepare the lipid bilayers. So the black lipid membranes we can prepare just by painting the lipid bilayer on a small pinhole using this special chamber. So this is the lab made actual chamber. And also we can prepare the lipid bilayer on a micro at the tip of the micropipette. And also we can paint the lipid bilayer. We can flush several solutions of lipids to prepare the symmetric lipid bilayers. And also the supported lipid bilayers we can prepare using conventional lipid bilayer preparation methods, and also the lipid vesicles. We can prepare the lipid vesicles. They have this ability, so we can tag the lipid head group. So we will have the fluorescence emitting lipid vesicles. And the microscopic technique we are using in our study is these here summarizing. So we are using the wide field fluorescence microscopy, and also the high resolution total internal refraction fluorescence microscopy, TIRF, and also the differential interference contrast microscopy and confocal microscopy. I'm not going to talk about this more, but I will explain like how we can use the patch clamp to study these changes, like how when the cell membranes interacting with these small molecules. So we all know that we can measure the electric current activity across the cell membrane by attaching this micropipette. So this we can do by getting this electric current trajectory. This baseline that reflects the undisrupted, non-leaking cell membrane, and the high electric current activity, the high amplitude, they reflect the disrupted, leaking cell membrane. So it's just that. We can do more analysis. So we actually found these small molecules. They are capable of increasing the electric activity of the cell membrane. So like here showing some of the trajectories, so they can cause electric leaking and electric increase in conductance states, like with higher concentrations they have higher conductance state, like that it's like concentration dependent. But the thing here is this is not just like two states. This is a complex process, like convoluted process. So they represent several intermediate states. So it's not just two state system as I explained, like non-leaking and leaking, but they have intermediate states. So this there's a classical way to analyze this BC fluctuation data using this autocorrelation function calculation. So we carried out this analysis to further study, and what we found was, interestingly, this we fitted this by exponential decay fitting. So this stochastic process, it's non-Markovian, meaning that's not a single well-defined rate, meaning it's like a distribution of rate. So that's because this intermediate state or like this internal state, that affect the influence its behavior. So that's why it's complex, and this indicates that the system's memory that's affecting its dynamic of the membrane stability. So stability changes like at different times. So we have discussed all of these in our papers, so you are very welcome to read them. Then that was cells, and these are the planar lipid membrane. So we move to the simpler version of the lipid membrane, because so we can make more like better conclusions. So we were able to see the leaking activity, the electric permeability is high even in the lipid bilayers. We observed this using the calcium permeability. We observed the calcium permeability is also high, the electric activity is also high, and also like this is another method, and here also we observed the by through pure lipid membrane. There are these small molecules leaking occur. So that means like so we can directly say that these conductivity changes and these permeability changes, they occur because of this distorted texture of the lipid bilayer. So then we wanted to check, we wanted to see in, we want to study this in, we want to get the molecular level details using this computational study. So here showing the one of the system, we place the molecule at the water level, and then we, these are some of the snapshots of this trajectory. And so they easily move into the lipid bilayer, but they trapped in the lipid head groups, and they make more interactions like again with the lipid head groups. So these interactions, they can cause more disruptions. We actually quantitatively got these interactions, what kind of hydrogen bonds, what kind of like is the methamphetamines, hydrogen and nitrogens are getting involved with the lipid head groups and the outside water. So yeah, they are making bonds, hydrogen bonds. And like here, what we wanted to see was we wanted to check how much of energy that one of these methamphetamine molecule needs to penetrate into the lipid bilayer, because I already mentioned that this lipid bilayer has different region, different polarity gradient regions. So depending on the polarity, they should have different energy barriers. They should have different energies to penetrate. So we checked, we placed the molecule at the top of the lipid bilayer, and then we drag it and we check the energy like that. We drag it, we check the energy. So like that we got this beautiful energy profile. And we found according to that they need these energy barriers depending on the polarity gradients. And also we asked this key question. We observed that this molecule is trapping near the lipid head groups. So we observed that also using the density profiles. Also we observed that that's where they are energetically favorable place to stay. That's why that molecule is staying below the lipid head groups. So then it's not just one molecule in our actual experiment. So we increase the number of molecules in the system. So like to 26 molecules and to 15 molecules, 50 molecules. So we saw that these molecules make in clusters. They like trapped at the lipid bilayer center of the lipid bilayer, and they make more interactions. They go out again, more chaotic. So then we increase the lipid complexity as well by introducing one more lipid, the cholesterol shown in green color here. There also we observed the same, and more interestingly, we got that. The water leaking, a water wire formation into the lipid bilayer center. So this can cause the lipid thinning, showing in here red color. So this can cause the lipid thinning. So the lipid density is getting thinner. So we also observed how the cholesterol is affecting, because the cholesterol has a more like a planar structure, so like they are rigid, and the DOPC, if we take DOPC has more loosey structure lipid in the lipid bilayer. So molecules would like to stay in a more loosey structure place compared to cholesterol. And also we observed that these molecules, they are not penetrating into the lipid bilayer as a whole cluster, but instead they move one by one as a single methamphetamine molecule. So that can cause the density changes. Because you can see some spots are darker, some spots are more green at different times. So more like green different times. So these results, these results showing like they can interrupt these lipid bilayers integrity and the density, the packing, lipid packing. So then we wanted to check how these molecules behave in, like how they are tumbling motion or the orientation, because this tumbling motion compared to this lipid bilayer normal, that means it reflects how well these methamphetamine molecules get adjusted to their surrounding environment. So for an example, like when the molecules when they penetrate through the lipid head groups level, it's stochastic, the dynamics, because it's experiencing drastic polarity changes. But anyway, this molecule, it's aligning with the surrounding lipid polarity. So I'm not going to talk all the numbers here, but we quantitatively got all of these observations. But the thing here is this molecule, it's never happy anywhere, because it's like this can interrupt the arrangement of the lipid bilayer. So then we wanted to check how well these molecules get dissolved in that particular environment, because when the molecules in the lipid head group, like the lipid, the extracellular water, it's different from the bulk water. So we want to check that, and also it's obviously different from when the molecules in the lipid hydrophobic region. So we check these. And so this is showing all the quantitative values, and these are like compatible with the literature data. But like what we have discovered is it's far more beyond these numbers, these quantitative values. This concept is more important, because like solvating in lipid and solvating in the extracellular solution, and then we can generate these thermodynamic cycles using that. Okay, so the best way to see these things, visualize these observations, is using fluorescence assays, the fluorescence imaging. So we use these the tagged lipid vesicles, fluorescent lipid vesicles, and also these are smaller unilamellar vesicles, and we also did this study to using the supported lipid bilayer to study the the lateral diffusion of the lipid to measure by measuring this fluorescence recovery after photobleaching a certain area of the lipid bilayer. So we can measure the lateral diffusion. So and this is the giant unilamellar vesicles. You can see the calcium. We saw the calcium permeability is higher when the methamphetamines getting involved, because they can entrap the integrity of the lipid vesicle. And if you think this lipid vesicle as a 3D ball, so when only the lipids are there, so it's just like swelled. But when the small molecules are getting into the lipid bilayer, so they can make this more like stiff. So here also we saw this, and we got all of these like technical conclusions. We made these technical conclusions by probing this combined technique to measure the permeability, stability, integrity. So, but here the scientific conclusion here is important, because this methamphetamine molecule, it's chemically distinctive molecule, and this is like we can use this as a powerful model system to study this like complex living environment. So because of this specific character, specific unique dipolar nature, because it's affecting its behaviors inside the cell membrane. So this is a valuable tool to understand most of these biological processes, the cellular processes in living environment. So I would like to thank my supervisor, Professor H.P. Tulu, and also all the funding sources, including this Ohio Supercomputer Center. That's where we do the all the computational studies, and my Department of Chemistry, Center for Photochemical Sciences, Bowling Green State University, and all of my lab mates, including previous lab group members. And so I would thank you all for listening to my presentation. If you have any questions, I would happily answer.
Questions, Hashini? Question here.
Thank you. Very nice and interesting talk. I've got a couple of quick questions, and can be partly from my ignorance. So, regarding your electrophysiological measurement, you're measuring current. So I want to know actually what this current is coming from. Who is carrying it? Is it the molecule itself because of being protonated, and or is it a momentary transient breakup of the membrane and then it causing, is it sodium? I don't know. What is the charge carrier?
Yes. So we are thinking of both because like not directly from the molecule, because they can interrupt the membrane, and then it can cause other ions to move. So we are thinking that mostly.
Because I was thinking if it's more like a compromising the membrane integrity,
yes,
I would have expected more heterogeneity rather than a binary suite, like a channel-like signal, you see what I mean?
Yes.
The second question I have, being very lipophilic, as you probably alluded to, like benzocaine.
Yeah.
In your simulation, do you see a lateral diffusion rather than really axial flux of this molecule moving from one end to the another, to the z-axis rather than in a y-axis, x-axis, whatever you say?
Well, we didn't check, we didn't study the lateral diffusion that much. But we did a study, like we got what is it called? I don't have that data. Yes, but we did a study. It is showing, but it was not that much change. It was not that much change, but it was change with the methamphetamine and without the methamphetamine. It was different, but not like a huge amount.
I more meant the lateral diffusion of the methamphetamine itself.
Oh,
Does it actually laterally move across rather than going to the z-axis? Sorry to take time here. So.
yeah. Sometimes it is, yeah, because it is moving everywhere, but it's not going through like very much far from where it's positioned. It's more stay into the middle because we place it in the middle. So the middle means at the normal of the bilayer. So yeah.
Thank you.
I had a quick question also from my ignorance. Do you think there's any information from people who take overdoses of amphetamine, for example, that they have such effects on their lipid bilayer, which could be toxic?
Yeah, I mean, it is toxic. Yeah, but here, like what we try to answer is like there are still unknown things about this. They disrupt these the synaptic vesicles. So maybe this is causing because of these trapping molecules inside lipid bilayer vesicles. Maybe that's the cause. So we cannot directly say this is toxic or not. We cannot directly say these are making huge pores, but there is an effect. Yeah.
Thank you. Thank you very much.
Thank you.